much for joining us here today, Jonas and I here today on our talks. So yes, I'm here today to talk about mammalian toxins and evolutionary rates within them. And we talking a bit about sort of what that means roughly, why I've looked at mammals particularly, and talk about some of the more juicier results in there because they're always good fun in that one. So without further ado, random mutations in your DNA are the currency of evolution. So these mutations are the reason why we have so many diverse species. It's the reason why we have so many so much variation within those species as well. And typically mutations are going to fall into two categories. So you have your mutations where if the nucleotide base changes but the amino acid stays the same, then those are called synonymous mutations. They make up the bulk of what you see in genomes these days, and they don't really change the genotypic or phenotypic signal you will see in an organism, but they are still present. However, if the nucleotide bases change and you end up with a different amino acid, or the sequence shifts or it stops, you have a non-synonymous mutation. And these are where it gets quite interesting because they can critically impact the fitness of the organism. That can either generate a new trait, or it can either generate a new variation in the present trait already. So and it's when you have different selection pressures acting upon organisms such as disease, predation, whether that be you're the prey or the predator, the availability of resources, or I think the most interesting one, which is how sexy are you? Very important bit of selection pressure that. These non-synonymous random mutations can become fixed within the population as they might provide a adaptation that increases the fitness of that organism. So that's when it gets quite interesting, and you can almost start to measure where these mutations when these mutations are much more prevalent within a certain gene, for example. So how would you do that, for example? You've got a gene and you're interested in how it affects, if selection is acting upon it on a population or a species level. The classic example here being the peppered moth, which I really encourage you all, if you're not familiar, to take a look at, and the way you can do that is by DNDS. So that is measuring the ratio of non-synonymous, so those really, so those change in mutation, the ones that critically impact the fitness, to the amount of synonymous, those neutral mutations. DNDS will, you calculate DNDS and that will give you a number. And if that number is more than 1, you have diversifying or positive selection present within that gene you're looking at, that there are much more of these non-synonymous mutations, that they are allowed to occur much more in the genes. Therefore, you assume that adaptations are occurring and there are selection pressures acting on it that might be encouraging these random mutations and these new, very different ones to occur. However, if you get a result that is less than 1 in your DNDS, then that means there is purifying or negative selection, and you assume that your genes are being kept stable, that you're not having these really dramatic mutations causing a critical impact in the fitness, for example. You typically see really, really low rates of DNDS in like housekeeping genes, for example. So why is this with Venom? Why does this matter? Enter this really, really juicy paper in 2015 by Kartik Sinagar and Yehu Moran, who I hope all of you are at least familiar with, or you've definitely read one of their papers, I guarantee it. QR code will take you to the paper, by the way, if you want to give that a quick scan. It's up for another slide or two. So the rise and fall of evolutionary innovation, looking at the different strategies of how venom evolved in ancient and young animals. So what they did is they took large toxin groups, such as elatrotoxins in your Latrodectus and Steatoda spiders, or three-finger toxins in snakes, and they examined what was the rate of DNDS occurring in that, and then calculated all the toxins for in a group. So what they found was that if you looked at ancient orders, such as Cnidaria, spiders, or centipedes, the amount of toxins that were acting under purifying selection was much more significant. It took up, say, 3/4 or more of the amount of toxins measured. Whereas in your younger groups, like your cone snails and your snakes, for example, the amount of toxins under diversifying selection was much more high, a much more significant chunk. So again, like 3/4 of them were under diversifying selection. So as a result, Sungar and Moran proposed that they're the two-speed model of venom evolution, and the idea being that when a species evolves venom and it becomes part of the fixed in the organism, there is this massive rapid diversification tied in with like diet and range expansion, followed on by a much longer period of purifying selection that preserves the toxins. So simply, you evolve venom and you just chuck the kitchen sink and see what works, and then eventually you figure out, separate, keep what is most important. And that's not to say, by the way, you're not finding lots of diversifying toxins within these older orders, nor do you not find any diversifying in the younger orders. It's just to say there is a general pattern of these. However, there were two caveats to the study. One was the venom they looked at was exclusively predatory, and they didn't look at mammals, which, as you might have gathered, I really wanted to do. So mammals, it is criminal how little there is on mammal venom. It's very understandable. A lot of these animals are nocturnal. A lot of these animals are elusive and just very difficult to obtain for various reasons. But venom can be found across a very unusual distribution in the mammals. So you have your solenodons and shrews and Eupotyphyla, vampire bats and Chiroptera, slow lorises and primates, and of course your platypus and your monotremata. You have 6 different functions identified within the mammals, which is the most so far of any group of venomous animals out there. So you have this unusual distribution, you have very unique delivery mechanisms tied in with very unusual patterns, such as seasonal breeding behavior in the platypus, so venom is only active during certain times of the year, or a 2-step model, such as in the slow lorises, who have to lick the brachial gland on their elbow before they can activate it with saliva, before they can deliver a bite. And you've also got these different usages of the venom. So as a result, mammals represent a really interesting place to look at the evolution of venom and examine, do they fit in with this 2-speed model? And if so, why not? Or why do they? So it's only now in the last, genuinely, it's only been in the last year, 2 years, that we have got enough data that we could actually attempt to start and answer this question. So realizing that, I went, okay, let's have a go at this. So overall aim is to identify the rate of selection acting upon mammals' toxins. 2 hypotheses. 1, that the overall rate is going to be acting on diversifying selection. And this is due to the fact that these mammals are generally, when you compare lots of venomous orders, mammals are still quite new on the block, let's be honest here. And also previous work has indicated that sometimes that some of these are actually already acting under diversifying selection. So that's why I got that hypothesis. And then the other one was that if there is difference in these rates, it's going to be down to the evolutionary age of the orders. So if I go back very quickly, I can just show you that you can see that mammals split into 2 in terms of the age of the order. So your monotremata and your Eupotyphyla are quite old, whereas your Chiroptera and your primates are much, much newer on the scene. Methods, just to say, I'm not going to go into heavy detail with these. If you are interested and you'd like to ask me a little bit more about what I've done and all that, please do drop me an email or tweet or ask. I'm very happy to talk about it. It's not that I don't want to, it's just that that will take 15 minutes of a conversation. So just going to focus on roughly what's happening in each. So first of all, first step was actually getting that data. Literature search, going through Uniprot, GenBank, you name it, you know where you find your venoms. That's what I had to go for. And the criteria that I had was that you needed at least 4 sequences represented in that toxin for it to be used. Align the sequences using Trim AI and Genius and all that jazz. This is where I mentioned that while I did the overall group, I also did some species-specific and order-specific ones, just because if there are any juicy results or some intriguing data, I could perhaps have a more specific look and look at maybe why that might be. Step 3, using DataMonkey Adaptive Evolutionary Server, which is really great. I recommend if any of you are interested in evolutionary studies, if you're not familiar, do have a look at the website. Great resources, all free on there. Fel, Fubar, Absarel, and Meme, those are the actual names, by the way. They're really great things. And what all these do essentially is they tell you where there is diversifying or purifying selection actually at a specific site, which is really interesting because then you can figure out perhaps if the overall selection on the gene is positive, but there's loads and loads of purifying sites. That tells you a bit of the story about the age of it, for example. Then to get PAML, which gives you the DNDS, that golden ticket we're looking for this work that I've been talking about, you need to do a maximum likelihood tree and then run it through that. And then finally, I'm not discussing any results, but Tree Sap is a really interesting bit of software, and I'm not sure how often people use it in Venom work these days. Again, do encourage you to look at it, because what it tells you is not only where there is diversifying selection at a site, it will tell you if that has impacted an amino acid property, which for the protein people out there, that's really interesting. So what are the results? So total amount of toxins, there were 8 toxin groups identified: Calichrome 1, Plasmagen activator, desmopilions, PCAP, CRISP, Kunitz domain 1, Kunitz domain 2. The vampire bats, particularly Desmodus roundatus, beg your pardon, is represented in all 8 of these groups. The Solenodon and short-tailed shrew are represented in the Calichrome 1, and the CRISP is where the platypus is represented. Sadly, there are no primate/slow LoRa sequences in there. And again, that's something I'm hoping to work on with my supervisor, Anne Karis and Vincent Nigel, because again, not due to lack of trying, it's been just so difficult to get actual sequences from these guys. So hopefully in the next few years, whether that be me or someone else, we're going to have some sequences to take a look at. So the actual selection rates, so those DNDS totals, well, really interestingly, it was as the hypothesis. We can tick that off. Most venoms are evolving under diversifying selection. So they are evolving with much more DN non-synonymous mutations occurring within them. The exception here being Calichrome 1. So I just want to focus on Calichrome 1 and CRISP to have a little chat about what is those results, because there was some interesting stuff in there, and that's been picked up by some really great papers out there at the minute. So Calichrome 1, as I said, I did some species specific ones. So I did one for Blarina brevicorda, which is the short-tailed shrew. 16 positive sites identified, and although it's still purifying, it's a higher rate of DNDS found within the calichren in the shrews. So these positive sites corresponded to inserted loops that were identified in 2009 paper, that while the Calichrome 1 was evolving under purifying selection, when they specifically looked at these inserted loops, they found that they were actually acting under very high diversifying selection, and that they proposed that this is where the toxicity of the Calichrome 1 is proposed sites of toxicity in Calichrome 1. Pardon me chewing the furniture over that one. But more intriguingly, a paper released last year by Agnieszka Barua identified that Calichrome 1 found in mammals and the Calichrome 1 found in reptiles, so Heloderma, our beaded lizards, is found on a shared syntenic locus that means that we've got at least one paralogous Calichrome 1 gene shared by these two groups. There's at least 300 million years of divergence shared between these two groups, and it's just so fascinating that perhaps Calichrome 1 is predisposed to be a venomous, used by venomous animals. It has this toxipotency, as they use in the paper, and just really indicates how old venom might actually be, and if it's an ancestral trait. Loads of excitement. Again, I really recommend checking out that paper. Happy to send it if anyone's interested. So CRISP. Again, as I said, the overall rate was 1.2923. That includes platypus and vampire bat sequences. But when you just looked at the vampire bat, you can see there's this shoot up in the how diversifying the results were. This is the difference in evolutionary age is one way to explain this discrepancy, but it's also worth considering what these animals use the venom for, which is why I want to talk about very briefly moonlighting, which is when you have a protein that has more than one physiological role within an organism. And this has been proposed for the platypus. So as I said, the platypus uses its venom in the males use it in the breeding season. So November to February, they're pretty chill. Then October to March, they get really horny and they try mating with everything. Wong et al. 2012 looked at the expression rates between in-season and out-of-season venom glands and found that CRISP was expressed at actually a pretty equal rate the entire time. CRISP has been implicated to be in immunity, so helping combat viral or microbial threats. And there was a proposal that while it is still an important component of CRISP, I'm sure you're all familiar, you've seen it in other venoms before, it also has this protection aspect when it's not being actively used for this very painful sting. And the vampire bats, I think, as we've all unfortunately found out in the last 2 years, great disease reservoirs, and that on blood feeding is so dangerous because you are exposed to so many disease threats. Vampire bats are also quite social animals, and they share blood feeds with people with vampire bats that haven't fed. So there's this argument that, again, this constant threat of disease is why you have this quite raised rate in the vampire bats. Also because in vampires, CRISP has been implicated to be vasodilatory, but there are other toxins that already fulfill that role. So there's this relaxed selection possibly that explains this different rate. So conclusions, I apologize, this is 50 minutes, but I am on the final bit. So just to say that the conclusion is mammal toxins are evolving under diversifying selection, fitting in with the cone snails and snakes. So a big tick. They do, as far as we can see with the data available, follow the two-speed model of evolution. However, whether that's to do with the evolutionary age is still quite difficult to say, because mammals have so many different unique selection factors acting on their venom because they use them for so many different functions. However, considering these functions is still absolutely fascinating because it really provides a window that we don't get to see with different animals. It's also lovely to be able to say that actually we can put venomous mammals really on a pedestal and put them next to other different venomous groups. Very often mammals feel like an interesting footnote in venom as opposed to an actual group worth considering. Future work, Silurises would be great, not going to lie about that. So not only data on species we know are venomous, but species that are putatively venomous. This little guy here is the Canarian shrew. One paper in, I believe, 1985 on a behavioral study, they chucked a shrew in with a tank with some lizards, and my God, it's undeniable that this is a venomous animal. And that's it. That was the last time anyone looked at them. So I hope you understand my frustrations as well. I hope today, if you take away one thing, is that these are animals so worth looking into. There's so much we still have to do. And perhaps if any of you out there are wondering what you can do for a PhD or postdoc, mammals, please be mammals. Thank you all so much for listening, and a special thank you to Mike for putting on the whole Venom Wednesday. I'm really glad to be able to speak to you today. Big thanks to my supervisors, Loris Lab, Katie Hudson, who helped me figure out panel. Feel free to follow me or drop me an email. And with that, thank you all very much for listening.
Yeah, so thank you very much, Leah.
Oh, no, thank you.
Are there any questions you want to ask Leah now about her mammal venoms?
Just leave it on the screen for a minute.
So I would have a question. So I would like to know, so in principle, if I understood it right, you are counting the mutations, right?
Yeah, it's about using the amount of mutations that are available. It gives you a rough idea of if there is selection and adaptation occurring. Yeah. So
and what is the reference sequence? So the original form, the plesiomorphic sequence, or?
Yeah, that's a great question. So with this, I'm just going to go back a little bit further. So with this one, there are different ways of looking at venom and how it's evolved, because you're right, one way is doing the is comparing it to a ancestral sequence, so to other non-venomous animals and mammals, and then comparing and seeing how it's evolved there. So what I've done is I haven't used any of the ancestral sequences. This is just looking at the venom exclusively, and this is what is done in this paper here by Sinagar and Moore, and they don't use any, they just use the toxin sequences associated. This is also why there's only a few venom groups I've used, because there's a lot of different toxins you guys might be familiar with, like the platypus, for example, has the defensins, which are some of the first sort of venoms in mammals identified. Couldn't include them, there weren't enough sequences. So there's no ancestral sequence, it's just looking at the mutation across these different mammals and the amount of mammal toxins produced. Are we seeing lots of changes all the time, or are they quite refined in what they're doing?
So it's a comparison of all the mammalian toxins. So you take a single toxin and compare all the autologs and
yes.
Okay,
yeah.
Okay.
So comparing across population, like different populations of Blanaria brevicorda. So it's from two or three different studies that I've been able to look at, and seeing that the rate is still quite suppressed is very interesting, because that means that this has been again, this is quite a refined the toxin, dare I say it, a toxin that's been in the arsenal for quite a while, worth considering, for example. But no, thank you for the question, Jonas, because that was a question thrown up by supervisors as well. They were like, could you just talk us through that? So I hope that explains the question you're asking about if I was comparing it to non-toxics or not. Just to say, if anyone would like to know that answer, join the club. And if you would like to help work with gene ontology terms or perhaps discuss about carrying that on, feel free to email me, because I actually have some data, but had to sort of leave it behind due to issues. So if you are interested in that, give me a shout. Just casually bringing up the whole, come have a chat with me. Actually, sorry, I'll go back to the beginning, but yes, no, thank you. Ooh.
And we have another question on the chat from Furriest. Do you also perform functional studies for the identified toxins?
Oh, functional studies, so identifying what they actually do within the mammal, what they do to the target organism. I have had to rely on the literature and see if they've done that. And I'm going to be honest, again, compared to groups like snakes, for example, that's something that's just not been done with many mammal species. Exception being is calichrome 1, which induces, it's a serine protease, so it induces, it causes a massive drop in blood pressure and causes paralysis in target organisms. And then for the other one that's been looked at is some of the vampire bat toxins. So the famous one, plasminogen activator, is used. It helps, if I recall correctly, it prevents blood from clotting. So it's actually been used in medicinal purposes. But unfortunately, for a lot of mammal toxins, it's a case of it being identified, but not being looked at on the functional level, just because. Well, great question. I don't know why they don't do it more often. Thank you, though. That was a great question. And again, that's something that needs to be done in the future.
Okay, then another question. According to slide 1, slow lowers preferably choose espresso martini.
Oh, yes. Sorry. Shall I go back to the beginning? That was a great spot, by the way. And I'm also glad that that's sort of, I've actually got the chat up here. Yeah. Preferably choose espresso martinis. I did try to pick something I thought these animals would enjoy drinking. A smorgasbord is not a charcuterie board, but hey, you know, so they put baby bells on there. Shrew got a pint, don't know why. Platypus is an Australian animal, so Foster's seemed an appropriate choice. Vampire bat, a glass of wine seems like something they would have. Espresso martini for the slow lorises, as I've got a number of friends and my supervisor are working on coffee plantations in Indonesia, and the best way to work with the farmers out there and also ensure the slow lorises are kept safe. And Celine Don has got a mojito there because they are from Cuba and the surrounding islands. It all seemed like fairly appropriate choices. I can't lie, I did spend a little bit too long probably on this, but nothing like a good introductory slide. Thank you for the pointing out of that.